Also, at this point, this is the maximum strength, which is the maximum stress that is measured by, that induced by this tissue before it started to break, to physically break. So this is what we call the strength of the tissue, and this is what the maximum, which is the maximum stress point. Also, we look at the area under the curve. This dotted region, this is what we call the resilience of the tissue, which is the area under the elastic region of the curve, and this one, the area here, represents the tissue toughness. So these are several mechanical properties that we could learn from this experiment, and this is what we had. So initially, these are the tissues that we generated. These are the control tissues, which is tissues without cytosine, without carbon nanotubes. It's made only of cells and collagen. And here we have with collagen and with different concentrations of multi-wall carbon nanotubes. And these are representative images for the stress-strain curves. As you can see, there are really very big differences. So if we look at details rapidly, we can see that with the multi-wall carbon nanotube, we have kind of increase in tissue stiffness, but only with this concentration of the carbon nanotube. The yield stress, or what we call the proportional limit, was increased by the multi-wall carbon nanotube. Also, the elasticity increased, the resilience increased, the strength increased, the ductility was increased, the extensibility, which is how much the tissue was extended before it break, and also the toughness. All these parameters were increased in the presence of multi-wall carbon tube, though the tissue has become more elastic, is a little bit stiffer, has greater resilience, greater extensibility, greater toughness, which means can endure more physical stressors. And this is actually what we really wanted to have. We made histological analysis for the tissues by using tritic phalloidine stain to stain the cytoskeleton of the cells, and also DABI to stain the nuclei. And we've seen that the cells were integrated within the tissue. They were viable. They did not die due to the presence of carbon nanotube. And the cells were organized in lines across along the lengths of the tissue, which means that the cells are functional. So they are there, they are functional, they are viable, and the tissue's mechanical properties are really enhanced. Now, these interesting results were also published in the International Journal of Nanomedicine. Now, to go further, now to check whether these tissues now can improve the healing of wounds, and therefore now we went to in vivo, and here we wanted to investigate the healing properties of these tissues. But here at this point, we thought of also incorporating a growth factor like angiotensin II, because angiotensin II has been shown to be involved in wound healing, not only in cardiovascular diseases and the regulation of blood pressure, but it's also involved in the behavior of fibroblasts in the skin. So we thought maybe adding angiotensin II could also be beneficial. Now here we generated in new tissues, but this time we generated them in the form of patches to be suitable for transplantation into wounds. And then we went to mouse model. One mouse model, we created identical wounds on either side. So one side will receive the treatment tissue, and the other side will be the internal control for this for the treatment side. So each mouse has its own internal control, so that we can minimize the individual variations. So the tissues then was wrapped with Tegaderm, this kind of polymer that is used clinically to protect the wounds. This was, of course, very important to keep the tissue in place and to prevent its dryness. Now, after 14 days, the animals were sacrificed, but for sacrificing them, we imaged the mice at day 0 and at day 14, which is the day of euthanization. So we have here the upper panel. This is for day 0, and the lower panel for the same mice after 14 days. So we have here the control angiotensin 2 group 0.025 with angiotensin 2, 0.1 and 0.1 carbon nanotube with angiotensin 2. We made macroscopic analysis for the wound closure, and these are the parameters that we used. So here the dotted red line represents the initial wound area. Then we have this blue line. This represents the non-contracted area, and here this is the contracted area because this one is not healing. So the one started here and then it ended here. But this is a normal skin. This is healthy skin. So why it's brought here? Because during wound healing there is a contraction process. So the wound normally, this is the normal phenomenon, that the wound contract to minimize the size of the wound. So this is the contracted area, and this is the non-contracted area. Also, we can see here this is a newly healed area. So this is kind of what we call epithelialized area, and this is what we have in the center is the open area, which is what remained from the wound and still open, is not healed. So from these main regions, we could calculate the percentage of the non-contracted area, which was calculated by dividing non-contracted area by the initial area, then the percentage of the epithelialization. Then we have, and for that we have the epithelialized area divided by the initial area, and the wound, or the percentage of wound closure, which is the closed area divided by the initial area. And what we had was really interesting. For the non-contracted area, it was all increased. So what does it mean? Normally, when a wound heals with contraction, this causes distortion of the skin texture and appearance, and it will leave scars at the place of the wound and marks that will stay forever at the site of injury. So wound contraction on one side is useful to accelerate the wound healing, but on the other side, it can disfigure the shape of the skin. So since we reduced the wound contraction, this was what we consider a positive effect, that this will reduce the scarring of the tissue and the tissue will heal with better outcomes. Also, the epithelialized area was increased, but here it was increased more or significantly in the presence of angiotensin 2. This is all we were lucky that we added angiotensin 2 in our study. Otherwise, we could have missed this promising effect. Now, angiotensin 2, we've shown that it increases the epithelialization. It increases the growth of the epithelial tissue, which is a very crucial step in wound healing. However, the wound closure at the end was not changed, let's say. The histological analysis for this transplanted tissue showed that there was no change in the thickness of the epidermis or the thickness of the dermis. This is good because we don't want it to be changed. We want it to be normal. We don't want to have a lump of tissue under the healing wound. So the tissue actually resolved within the tissue. It integrated very well, and the excess tissue maybe was resolved somewhere. So the histology was in general normal. So in conclusion, of course, this work is submitted to a journal recently, and we hope to have good news very soon. So in conclusion, the engineered dermis tissue containing chitosan and multi-wall carbon nanotube have improved the biomechanical properties of the tissues, or the EDTs can improve the quality of wound healing in terms that it enhances wound epithelialization and reduce wound contraction. And at the end, I would like to thank my group and colleagues in our university, and I would like to thank you also very much for your attendance and hearing.
So thank you, Dr. Naeem, for the nice intro presentation. And I think it is a lot of research efforts, and it's really outstanding and remarkable research at Najaf University. And now it's time for discussion, and I would like to. So if you would like to speak in the mic, just raise your hand, or you can write your question in the chat room. Um, so yes, we have in the chat room question, Dr. Naeem.
Okay. Um, in the chat room.
Yes.
Oh, yes, I found it.
Shall I read it to you or?
Yeah, yes, please. I would be grateful.
Okay. So are multi-wall carbon sterilized before use?
Yes, yes. That was a very important point. The carbon nanotubes were put under high energy UV light and was steered continuously so that it will be exposed to this UV light for sterilization. In addition, after we combined it with chitosan, the hydrogel was autoclaved under certain programs, so we have a sterilized product.
The second one, can infection or inflammation occur after using multi-wall carbonate
Yeah,
nanoparticles
this
on
is
bonds?
yeah, this is very important question. Actually, accidentally we had events that we found some infection in certain mice, but this was kind of scattered event. So it was not kind of a trend, something that comes up during the procedure. So in all experiments, we had maybe one or two infection cases. So it was not, we could not attribute it to the treatment. Also, I would like to remind that the chitosan, which we have in our preparation, has antimicrobial activity, plus that we try to take sterilization procedures so that we minimize this thing. Now, regarding the inflammation, the histological analysis did not show any signs of abnormal inflammation. So that was kind of a normal level of inflammation that could be associated with the healing wounds, but not something extra or something unusual.
Do you have any further question? You can write your question in the chat, or you can raise your hand if you'd like to speak with your mic. So I have a couple of questions to you, Dr. Naeem. So the combination of schistone and a multi-wall carbonate nanotube, you used like 0.1 at the concentration. Do you expect if we increase this concentration, can we get better results for wound healing?
We actually tested much higher concentration than we used, but the result was toxic to the tissue. So the cells died and the tissues did not form. So we reached to this concentration range after kind of optimization steps until we determined the range that could be safe for the tissues.
Okay, I got a second one. I noticed that all the experimental was on mice. Did you test any other animal experiments like, I don't know, monkey for instance, collaborations or I mean, I am curious to know about the different response of different animals to this treatment.
Um, Thank you very much, Ahmed. Actually, I have to be honest. I have to talk about a disadvantage in our model, that the healing process in the mice are somehow different from that in humans. So the wound contraction, the scarring formation in mice particularly are much less than in humans. So such wound if we that we have shown you in my presentation, if I induced it in human, it will produce very tremendous scarring, but in mice the scarring was barely noticeable. So I have to admit that our model is not, let's say, optimum for this experiment. But on the other place, it's not easy to conduct such experiments on larger animal models due to lack of resources mainly. And of course, we cannot move to human right now because there are lots of limitations for that.
Yeah, and I think wounds are different in the way that they used to heal, right? Or
Again, please.
the wounds differ in the way of healing, right? Or
Yes, yes,
or,
yes.
or all wounds that occur on the body have the same mechanism for healing.
It depends on the cause of the wound. So if it was a chemical or heat-induced
Yes.
wound, let's say, then the healing process will be different, and the challenges will be also different because you also destroyed the margin of the wound. So you have the wound, and also the cells on the margin are also injured. So the healing will be much more challenging. But in our case, we use kind of we use just a normal one. So we just excised a circular piece of skin. Now, if you are asking whether if the place of the wand, let's say in our experiment we used it on the back. So if it was, let's say, on the abdomen, would it be different? I don't think so, because at the end it's the same skin, it's the same let's say way we have an incision, but of course the blood flow in the tissue region could be different, and the blood flow is important for adequate wound healing. And as we know, that blood flow to the skin is not uniform, is not homogeneous. So part of our skin receive more blood than other parts. So we expect to have different healing pattern. But in our experiment, we use the same place always.
Yeah. And do you have like any genetic experiments like gene expression analysis to identify genes responding to this treatment so can like produce cells for fast healing or something like that?
This is something we are working on now. So the project is not finished. We are now in the process of another phase of the study that we will go with a deeper insight. So we will have studies at the level of protein expression and gene expression from Western blotting and real-time PCR. And for that, we will test for the expression of certain inflammatory mediators and growth factors that are crucial for wound healing. And we will try to make associations between these factors and proteins with the extent of wound healing.
Actually, this will be kind of interesting to know. Maybe next year you have like
Yes.
some results that can be shown in next MGBS.
Yeah, I hope so. We are working hard for that now.
Yeah. So, do you have any further question for Dr. Naeem? I think he really presented very nice results and research. Yeah, I think we don't have any further questions, Dr. Naeem. So I would like personally to thank you again for presenting this research, and I'm quite sure that all of us learn a lot about new techniques and technology for healing the wounds. And I would like to thank the attendees as well. And see you next week in new MGBS lecture. Thank you, Dr. Naeem.
Thank you very much. Thank you very much, and thank you very much for all the audience. Thanks a lot.
So just for the attendees, please don't forget to register if you would like to issue a certificate of attendance at the end of MGBS seminars. Thank you. Goodbye, Dr. Naeem. Assalamu alaikum.
Bye bye. Walaikum salam.